So yes, we'll now move on to Kate French. She did her PhD in the MIT Woods Hole Oceanographic Institution Joint Program. Before that, she did a bachelor's in chemistry from Yale University. And she's also done postdoctoral research at MIT and at HUI as well. And Kate was, I think, one of the first of the cohorts to participate in the RGNO back in 2014. And today, she's going to be talking about an aspect which was part of the thing that drove her curiosity to come to Namibia back in 2014, which was looking at potential evidence for anoxygenic photosynthesis in the Benguela upwelling system. So with that, I'll hand over to Kate.
I'm going to talk about pigments preserved in sediments underlying the Benguela upwelling system and whether they can provide evidence or not for anoxygenic photosynthesis. So the keywords are on this slide. I'm going to just go over them briefly. So biomarkers are biomolecules that can preserve over some time scale, and they provide biological and or environmental information. Phototrophic sulfur bacteria can use reduced sulfur to perform photosynthesis, but they don't produce oxygen as a byproduct. And we'll be talking about green sulfur bacteria and purple sulfur bacteria, which are two types of phototropic sulfur bacteria today. So sulfide is one of the reduced sulfur species that can be used by phototrophic sulfur bacteria. And it also is a key agent for organic matter preservation. And there's several features about the Namibian shelf that are of interest for this work. So one is the upwelling system, which is the focus of RGNO, but also are these really kind of unique sulfide eruptions that occur on the Namibian shelf, as well as the active sulfur cycle. And then we just heard a little bit about the organic rich sediments that are on the Namibian shelf, and those are also relevant to today's talk since that's what I sampled. So there's several well-known pigments, and you probably are familiar with some of these. And these pigments are what is giving a lot of the color in this image. So as an example, we have lycopene, beta carotene, and those are supposed to be pretty good for you, and you can find them in colorful fruits and vegetables. Also of you're probably familiar with chlorophyll, giving the green color to the plants that you see in your everyday life. And so chlorophylls are part of the molecular machinery that enable photosynthesis, particularly oxygenic photosynthesis. So that's using water, CO2, to harvest light using chlorophyll, to build biomass. So that's organic matter. And as a byproduct, these organisms produce molecular oxygen. However, there are many different kinds of chlorophyll structures. And some of them are universal, that's chlorophyll A. However, other chlorophyll structures have some taxonomic specificity. So you find them only in some groups of organisms rather than others. And these structural modifications of chlorophylls allow organisms to harvest light in different regions of the light spectrum. But there's another kind of photosynthesis. There's anoxygenic photosynthesis. And in this kind of photosynthesis, a different molecular machinery is used, and that's called bacterial chlorophyll. So similar to chlorophyll, however, again, some structural modifications. And as an example of anoxygenic photosynthesis, reduced sulfur, so in this case sulfide, can be combined with CO2, harvesting light to power that reaction using bacterial chlorophyll to build biomass. But oxygen is not a byproduct in this reaction. And different bacterial groups can do anoxygenic photosynthesis, listed here. And like chlorophyll, there are many different bacterial chlorophyll structures, again, with structural modifications allowing absorption in different regions of the light spectrum. And chlorophyll, bacterial chlorophylls, fragments of these can be preserved over long time scales in sediments. So here are the structures you would find in cells. When cells degrade, these pigments also degrade. You can preserve different forms of the ring structure. You can break the ring structure into smaller rings. You can also preserve parts of the chain of chlorophyll and bacterial chlorophyll. And then you can link these fragments back to the original structure, and then use knowledge about which organisms use a given chlorophyll or bacterial chlorophyll structure to then say who is growing at a given time. So in that way, these fragments of chlorophyll, bacterial chlorophylls can serve as biomarkers. There is another class of pigment that's of interest for today called carotenoids. And these are C40 isoprenoidal compounds, and they are hugely structurally diverse. There are hundreds, actually over 1,000 different kinds of carotenoid structures. And they can assist in the photosynthetic processes, again, for light harvesting, but they can also be used for photoprotection and to impart color. And in these images, I have two examples of carotenoids in the wild, I guess. And so we have purple sulfur bacteria in both of these, as well as this little green bleb, which is from green sulfur bacteria. And these will be the focus of today's talk. So in this schematic, I'm trying to illustrate how carotenoids from green and purple sulfur bacteria can serve as biomarkers. So on the left, I have a typical environmental scheme in which you may find these bacteria living. So they need sunlight, they need reduced sulfur, and they tend to live in anoxic environments in order to perform photosynthesis as described earlier. And they have different light requirements. So some need lower levels of light, some need higher levels of light. And so you may find them stacked in this kind of ordering within a water column or even sediment that has some sunlight penetrating. And they each have their own unique carotenoid that is associated with them in order to perform photosynthesis. And these carotenoids can be preserved over very long timescales in sediments. And they lose double bonds over the course of diagenesis, but we can still recognize what their original structure was and therefore who they came from. And in this way, they can serve as markers for the existence of green and purple sulfur bacteria in environments, as well as the environmental conditions that sustained them. As far as my PhD work when I was at MIT, one of the things that I found in collaboration with co-authors was that green and purple sulfur bacteria carotenoids are far more prevalent in geologic time, which is going down in this figure, than was expected. Before the study, these were generally associated with extreme events. So mass extinctions, anoxic events, mostly due to the physiological requirements, so sulfide, anoxic environments. However, when you looked throughout geologic time, you find that their occurrence is far more frequent than we expected. And that's especially because we don't think of green and purple sulfur bacteria as inhabiting today's marine systems. So this was a little bit surprising. And so how do we explain this ubiquitous occurrence in the geologic record? So several explanations are illustrated in this slide. So one possibility is that they could be living in mats, so within sediments. So it's not reflecting water column conditions necessarily. Maybe their occurrence in marine organic matter really has to do with basin configuration. So these are restricted basins, something like the Black Sea, where we know that they live today. Transport may play a role where they're being transported from coastal environments where they are known to live in today's world out offshore. So that could be a key effect that needs to be accounted for. Another possibility that had been used to explain the occurrence of these biomarkers was that Maybe they reflect upwelling systems that become transiently sulfidic. However, there was no evidence of this in today's ocean, and this is what I wanted to test when I went to RGNO in 2014. So the Namibian upwelling system seemed like a really good target for this because it seemed possible that it may have some of the key ingredients. So sulfide being one of the hardest ones to meet in the modern ocean. And we can see sulfide erupting out of the sediments into the water column in these NASA satellite images. So this is over the course of several years, 2007, 2014, 2015, and this is a natural feature of the system. And so we want to understand whether anoxygenic photosynthetic bacteria are part of the Benguela upwelling system in order to, one, understand the ancient sedimentary record, but two, also better understand sulfur cycling in the upwelling system. Are these key participants for processing sulfide in the upwelling system? So during RGNO in 2014, I was lucky enough to be able to join a week-long cruise on the Mirabilis. So I want to thank all the crew members as well as instructors who made this sample collection possible then. And so during that cruise, we visited a number of stations between Walvis Bay and Lüderitz, so 23 degrees south to 27 degrees south, mostly on the inner shelf, as shown here. So I collected samples marked in the stations in red. Although we visited the stations marked in black, I didn't collect at those stations. So most of the sediments that I collected were from water column depths, or were from sediments that were at water column depths of, 40 to 100 meters, although there were a couple that were even deeper than that, 100 to 200 meters. And so these images show sediment cores as they're being collected off the Mirabilis. And I collected mostly within the top centimeters of the sediment cores, as well as sediment grab samples when coring wasn't possible. The sediments I collected were frozen in the dark immediately after collection, and then all the way back to transport to MIT, where I worked on them further for pigment analysis. So this describes a little bit about the pigment analysis, and I used a library of reference material that I had collected in order to identify pigments from anoxygenic photosynthetic organisms, as well as anoxygenic photosynthetic bacteria. And then I used a sensitive analytical method to look for these, and these were the results. This is a figure out of my thesis showing that what I found was mostly pigment evidence for oxygenic photosynthesis. And so that can include cyanobacteria and algae such as diatoms. This is not very surprising given what we know about the system. I did not find any pigments that were diagnostic for anoxygenic photosynthetic bacteria. And this was kind of the end of the story. This was a thesis chapter, and then I just graduated. So that's where it had been left for a number of years until colleagues at MIT did follow-up work. And so I want to thank them for their contribution here. So we have Jian Ma, and we have Xing Chen and Roger, who's also on the call right now. And so I just want to add this slide as some context for their follow-up work. So sulfide has a dual role here. Not only was it needed for the photosynthetic process, it's also a key diagenetic agent. So it can sulfurize organic molecules like carotenoids. It can basically capture molecules like carotenoids into macromolecular organic matter. It can also reduce double bonds and carotenoid side chains. And so all of these reactions suggest that you really need to look at other organic phases than what I looked at in my thesis work in order to look for these carotenoids in a more complete way. And so that's what Jian and Xing Chen did. They looked at both nonpolar hydrocarbon phase, which would get at this kind of family of carotenoid degradation products, as well as carotenoids released, or other hydrocarbons in general released during Raney nickel desulfurization. So you can cleave out different structures that have been sulfurized, and you can look at them using a technique called GCMS. In this case, highly specific, highly sensitive technique using tandem mass spectrometry. And when they did that, they found an array of hydrocarbons in both the nonpolar as well as the desulfurized nonpolar fractions. And I'm not going to go over all those hydrocarbons other than to say that one of the families of hydrocarbons that they found were, in fact, carotenoids related to green sulfur bacteria. These were more abundant in the desulfurized phase. And more specifically, the pigments that they found were related to the low light dwelling strain of green sulfur bacteria that we find in the Black Sea today. These were in a higher abundance within the inner shelf, so stations 4 and 10 on my map in the earlier slide, compared to the outer shelf where they were more trace abundances. And this makes sense with respect to how we understand sulfide eruptions on the inner shelf. They usually are initiated on the inner shelf and then they get advected offshore. And so that matches the satellite observations. So there are a couple different possible scenarios about how we can understand this result. The first may be that the green sulfur bacteria are dwelling deeper in the water column where the bottom waters are sulfidic, yet light can still penetrate. This seems possible at water column depths of between 40, 100 meters, that you may get enough light penetrating into those sulfidic bottom waters. Another possibility is that they could be growing on sediment surfaces where there's both sulfide, and this is only possible if there's enough light penetrating down to sediment surfaces. Another possibility is that they are growing rather transiently, so only during sulfide eruptions where sulfide is emanating out of the sediment into the water column, and they find some sort of sweet spot where there's both reduced sulfur as well as light. And so these are all different possible scenarios for how green sulfur bacteria could be growing in the Benguela upwelling system. And so to summarize in a little bit of a future outlook, that we did find using different preparation and analytical methods that there are sedimentary carotenoids that suggest that green sulfur bacteria participate in the sulfur cycle of the Benguela upwelling system. It explains several aspects of the ancient sedimentary record. One interest to me is that in some sedimentary horizons, you can have evidence for oxia, dysoxia, while also having evidence for more extreme conditions such as sulfide in the photic zone, like the carotenoids from green sulfur bacteria. So how do you reconcile these? Transient events like sulfide eruptions may be one way to reconcile these two different lines of evidence within a given sedimentary horizon. So future work may be trying to use carbon isotope data for additional confirmation that these carotenoids really are linked to green sulfur bacteria. And of course, it would be really useful and exciting if we could get direct evidence of active phototrophic sulfur bacteria in the Benguela upwelling system. And then we could answer more specifically whether they're growing within the water column or sediments, and whether they really require these transient sulfide eruptions in order to grow. So I want to thank everyone again for making this work possible. That includes both the RGNO instructors, crew of the Mirabilis, as well as my MIT collaborators for following up on this work. And with that, I will open up for discussion and questions.
Thank you, Kate. Now you opened already the discussion. Are there any questions? Well, you would say now, if you could do it again, you would sample probably closer to shore in areas that are more often affected by sulfide eruptions, right? And then you would collect samples in the water column, just to prove your last point.
Yes to both of those. Probably would sample at some more shallow depths, and would also, it'd be good to get some water column samples. It might be a challenge to be sampling at the right place and right time, if sulfide eruptions really are key to this story.
Mm-hmm. Right. Well, you wouldn't have to go that far for that. You could go to Woods Hole and sample Salt Pond, which has
that was actually one of the reference materials I used for the HPLC work, to compare the compounds we found in Namibia with those, since we know that they are growing there.
Exactly. Or you could come to Lago di Cadagno, where you see the carotenoids as well. Anyway, that's a little propaganda for the nearest site. We could do this. But other questions? You know, when you mentioned the geologic time scale, I was wondering, these were times when oxygen was already long since present. Right? And so that means the ocean depths, like the Black Sea today, would have been very sulfidic. I would like to know, at what time in the geologic time scale did you identify these carotenoids that are linked to anoxygenic phototrophs?
Yeah, let me just pull that slide back up. So most of the slide is actually showing the Phanerozoic, which is the last 550 million years ago. And then just this short little bit at the bottom is the Precambrian. And so most of the Phanerozoic, particularly Mesozoic and younger, the oxygen in the atmosphere should be comparable to what it is today. And so, yeah, the question is, if the atmospheric oxygen conditions are similar to what it is today, you know, post 250 million years ago, how are these so prevalent in marine organic matter, since we don't think of them as being rather prevalent in today's ocean? So basin restriction is one possibility, and I'm sure that that's part of this figure, something like the Black Sea. But the question is it everything?
Yeah, so it's interesting to see how the story is developed, Kate. For the sulfur linked compounds, was there a sense as to what? So I remember there was some work done a long time ago on, I think it was on Peru margin sediments, where they were looking at this question of sulfurization of organic matter, and they could see that, yeah, they found certain carotenoids were very well preserved through this sort of association with sulfur. So
Yeah.
is there a sense how abundant these compounds are in sort of absolute terms in the Namibian sediments relative to other components that are present?
I don't think that, yeah, we have that kind of quantitative sense yet. At this point, we're just looking for kind of a yes, no, are they even there? However, they are a lot more trace abundance, just based on kind of noise to signal that we see in the chromatogram out on the outer shelf. And I think one interesting question is whether this is transported off from the inner shelf where the sulfide eruptions are more prominent. And so then you see some traces of that actually out on the outer shelf, and depths of 200 meters. So yeah, whether that's kind of relic material.
And then this is sort of a separate question, but there's a lot of discussion these days about these sort of cryptic elemental cycling, I guess.
Yes.
We call it. And so, I mean, could it be the case that these organisms are there, but you don't necessarily have to have significant ambient abundances of sulfide for them to be growing, so that there's a turnover that's taking place such that they can exist, but we don't sort of see the manifestation of that in terms of abundances of the things that we like to measure?
Yeah, I mean, I don't know what the sensitivity of, omic techniques are, where if you apply that, if you miss them, because they're kind of there in the background. So I'm not, yeah, I'm not really sure as to how difficult it would be to, I guess, get evidence of their activity or not, actually in the system, whether that's sediments or water column. Jake may be able to comment a little bit more on that. And so it may depend on, exactly, how they're growing, whether they're kind of in the background of, in the water column all the time, or they, kind of take off during these short-lived events, these sulfide eruptions.
A quick question. I was wondering if, sort of in line with what Tim was asking, if perhaps other organisms might have these carotenoids as well, and if there are genes that are specific to these carotenoids, then we can like kind of search like genomic databases to see if they're present elsewhere.
Yeah, so this is work that some folks in Roger's lab, including Xing Chen, have been doing. And so their recent work has shown that some carotenoids, not the ones that I've specifically showed here as part of this talk, but there's some other carotenoids, reniaritine, reniara purpurane. These can be produced by cyanobacteria. And one of the ways they found this was using methods that you've described. And so those were also found in the Namibian sediments and are probably linked to cyanobacteria. But as far as I know, isoraniaritine is still in association with green sulfur bacteria, and not so much with cyanobacteria as are these other carotenoids. So I don't know what future work will hold as to whether that linkage holds up, but as far as I know, it still does. Yeah.
More questions? Well, just as a last thing, maybe, the H2S has to be there, has to be anoxic, but that's hard to achieve because we always have the oxygen diffusing down. Yeah. So to find that very narrow band, is probably critical. And yeah, exactly. It's not stable in a marine situation like the Namibian shelf. So to find those is hard.
That's the advantage of looking in sediments, because it integrates all of that.
Mm-hmm. Okay, does Bronwyn have a question?
No, I just find this totally fascinating. Have any measurements been made of the actual light penetrations at these various depths?
Yeah, so that's a good question, and one that I was trying to follow up as part of this work. And so there, I believe in Mark Horn's thesis, he published on some of these stations and some measured depths of the photic zone. And so, the photic zone is clearly going to be dynamic in this environment. Very. So, he measured that in, I don't know, the 2000s, and how variable that is from season to season, day to day, I don't know. But yeah, the depths that he reported are definitely plausible that that enough light penetrates far enough into the water column, maybe even to the sediments, to support one of these models. One outstanding question for me is, clearly the photic zone is going to be really dynamic during sulfide eruption events where you have elemental sulfur precipitating out. So how the photic zone responds during one of these events, and how that intersects with sulfide concentrations, I have not seen that yet, but that is a key part of this.
Yeah, it's incredibly dynamic, and for the most part, we wouldn't expect light to penetrate at all to those sort of depths, not even to 100 meters, but it varies, as you say. So everything would have to be done at the same time. Mm.
Yeah.
Super.
Thanks.
Okay, then we'd like to end here. Thank you for participating. We will follow up on this because Yan Ma will follow up on his work a little bit, see how we handled it, because he He's in Shanghai, and we are at 3 o'clock in the afternoon. That's around midnight for him. But we'll see how we do this in a short presentation in the showcase towards the end of the seminar then. But thank you, Kate, for putting the framework for his presentation as well. And thank you for participating. Hopefully you can have an opportunity to continue with this type of work wherever you are now.
Yeah.
And come to Namibia once again if you need more.
That would be great.
We'd like to go there anyway, as soon as possible.